In this video, we will teach you how to upload, pre-process, and normalize expression data for mode. Data upload is performed in the Multiomics application portal. First, select the Data Upload tab, then specify your molecule type. These can be peptides, proteins, metabolites, lipids, or transcripts denoted as RNA-seq. Name the project and select Use Project Name. Next, we will upload an expression data file, which can be a CSV or text file. In the expression data, one column contains the biomolecule identifiers and the other columns contain the relative abundances per sample for mass spectrometry, the peak area for NMR spectroscopy, or counts of transcripts for RNA-seq based omics. To the right, we have our peptide proteomics dataset. The peptide column indicates the identifiers for the biomolecule of interest, and all the other columns contain the samples with relative abundance measurements for each biomolecule. Select Save Project and Submit to Server. From the Multiomics application portal, we can verify our omic project of interest is in the Manage Files tab. Here we can see the expression data we just uploaded. From here, we can go to the Map Store and select Mode. In the drop-down menu, we will select our project we wish to use. When we launch the app, if it is expression data only, it'll take us to Format Data tab.
We will cover how to carry out the pre-processing that mode requires for visualization. In the first drop-down, select which column contains your molecular identifiers. Next, if a sample information file was not uploaded, assign groups to samples by switching the toggle from No to Yes. Here, in the text box, enter the names of your groups, separated by a comma in order. For example, if you had an experiment with a control and 2 infection conditions, you may type control, infection A, and infection B. Make sure the number of entries matches the number of columns in your expression data file, minus the one column that contains the molecular identifiers. Click Confirm when ready. Next, a table will appear under sample data with the sample names and the group designation. If you would like to remove a sample, leave the group set to NA. The next few options are only applicable if the expression data are abundances from mass spectrometry or nuclear magnetic resonance, as opposed to RNA-seq transcript counts. Here, denote what value represents missing data in your dataset, which is typically NA. Set the data scale. Currently, raw intensity, log base 2, log base 10, and natural log are supported. If the original data scale is raw intensity, select either log base 2, log base 10, or natural log. Data will then be log transformed by this specification. We will show you how to properly conduct the data normalization step, which is only applicable to mass spectrometry or nuclear magnetic resonance data. If you have RNA-seq data, you may skip this step. First, select whether your data have been normalized already or not. If you'd like to skip normalization, switch the toggle to Yes and click Confirm. Otherwise, leave the toggle at No. Next, select your desired subset and normalization function. For more details on these parameters, see our video on the PMART normalization page or read our PMART publication. Both links are in the video description. If groups have been supplied in the sample information dataset, you may click the Test Normalization button to get a p-value from the Kruskal-Wallis test. This value will inform you if your selections have led to normalization factors that vary by experimental group, which can lead to biased downstream statistical results. A p-value less than 0.05 is not ideal, and we suggest trying another normalization approach. When finished, click Confirm to move on to the design plot step.